camel. I wrote to the camel after I left Jordan, and he never wrote back. It hurt me, but I've gotten over the camel. Well, thank you all for coming here. We at Salk greatly appreciate your interest in our institute and for coming to this wonderful music series. It's really great. So I am interested in how cancers begin and how they can be prevented or reversed. And I'm a molecular biologist, I'm not a cancer biologist, so my point of view is one of mechanism, and I will try to tone down the mechanistic aspects because I know that people can only take so much of listening to a scientist. So I've had my two best friends really help me with this to tone things down. So let me proceed. So how do sporadic cancers arise? Most cancers are sporadic. Most of us don't have inherited unfortunate mutations that make us susceptible to particular forms of cancer. So most are sporadic. And what is really striking to me is the very different distribution of specific cancers worldwide. So for example, breast cancer is highest in North America, and basically the English-speaking countries, Australia, New Zealand, and parts of Europe, and with a very low incidence in most of Asia and most of Africa. That's breast cancer. And what about all different types of cancer in a comparison between the US and Japan? Well, we see that prostate cancer is 4 times higher in the US than Japan. Lung and breast cancer are about 2 times higher. Melanoma is a whopping 28-fold higher. And then some cancers are higher in Japan than in the US. So already this tells us that sporadic cancers are largely determined by environment and lifestyle. And another striking illustration of this is the existence of cancer villages throughout China. So these are sites of where cancer of a particular form is very prevalent, and it depends on the type of industrial pollution that is being dumped into these various regions. So this is a very stark illustration of how our environment is the causative agent of specific cancers. So it's how we live and what we're exposed to. And as we know, certain excesses are associated with different types of cancer, such as excessive smoking and toxin inhalation. All of these numbers pertain to US cancers. Breast cancer is a little harder to assign a causative agent to, but it's definitely hormonally sensitive and diet influenced, as is prostate cancer. And liver cancer is associated with excessive alcohol intake and toxin ingestion. So most of us know not to overdo it with any of these causative agents, but unfortunately damage occurs to our cells, our tissues, as we age. And this is due just to normal living, eating, breathing, living a normal life. We create metabolic byproducts due to oxidative damage, depending upon what we eat, if we eat anything. Inflammation, of course, influences cancer, and we're all exposed to some extent to environmental toxins. So as we age, we start to accumulate damage to our tissues, which never may become, usually does not become cancerous, but certainly these cells are primed and vulnerable to increasing tumorigenesis. Sorry. Now, I found this striking when I first saw it, probably because I was over 40 when I first saw it. So this is the age-dependent incidence of cancer calculated for males over all types of cancer, but it looks largely the same for females. So things are going along really well until you're about 40, and then the curve really rises dramatically around 45. Once you're past 80, the curve drops because an 80-year-old is a very strong person. This person has good genes and a great immune system, great repair processes, so congratulations to anyone who's 80. That is a strong, tough person. But for the rest of us who are under 80, when I passed 40 some time ago, I thought, well, it's time for a lifestyle change. So what can one do? One can change their diet. Let's eat a lot of this, little tiny glasses of red wine, but lots of broccoli. And of course, the companion animal is very important. This is Duncan, he's great. So one can do things to try to help themselves, no matter what environment they're living in. And if you're a female, you can always do this. I don't know what men do, but I wish you the best. So, but fortunately, all of us should do these things, these preventive measures. Preventive health is very important. But otherwise, our bodies have very potent cellular processes that repair our damaged tissues to prevent them from becoming cancerous. So we are lucky that we have very good physiological systems. So let's just take a look at how cancer progression could occur. This is our normal tissue, and it is subject to acute or chronic stress all the time. So when stress happens, say in the form of a sunburn, which we've all had, that automatically activates our damage response pathways. So these are processes in our cells that quickly recognize damage or stress and try to repair the damage, try to return our cells to normal. And it activates a number of genes that I have worked on. So now you've got a damaged tissue, but it's not too bad, and your repair processes will either, your damage processes will either repair the damage, or if the damage is too bad, it will eliminate the cell. So that's good. That's what happens most of the time. However, if this isn't a complete process, then what you have are persistently damaged cells that remain in your tissue. And as I said, these accumulate with age, and we all have them, and apparently they're not all eliminated. And that's fine because they don't all become cancerous, but one can imagine that with further acute or chronic stress, these are the cells that can become cancerous. So our interest is in studying these precancerous cells. You can find them in normal breast tissue. So you don't find them by looking for cancer. You have to use specific markers that are very sensitive that tells you this is an abnormal cell. It's not cancer, but it's abnormal and it's stressed. So we are interested in how these are created and whether we can repair them. So remember, abnormal cells, whether cancerous or not, are stress cells. We tend to think of stress as psychological stress. Actually, our cells and our tissues are stressed too. So just as we don't want to be stressed all the time, if we're stressed, we want to calm down, we want our cells to calm down too. So our question again is, how do abnormal precancerous cells arise? And our experimental approach, all scientists have to have a question and an approach to answer the question. And ours is to isolate normal human cells from disease-free tissue and analyze their response to damage. So we deliberately damage normal tissue and monitor their recovery from damage or their conversion to a precancerous state. Now, you may wonder, if we use normal human cells, normal human tissues, where do we get the tissues from? Because no one wants to give up their normal human tissues. I certainly wouldn't. If someone asked me, if my dearest colleague asked me for my tissues, I'd tell him or her to forget it. So where we get this from, where is the source of normal human tissue? One source is from voluntary reduction mammoplasty. So many women undergo voluntary surgeries to remove part of their breasts for various reasons. They don't have cancer, but they're removing part of it for other reasons. And so we work with the Moores Cancer Center and receive tissues that are, of course, anonymous. We don't know who the donors are. We receive small pieces of normal tissue to perform our work on. So using these small tissues, again, not the whole breast, but a very small tissue, let's say the duct, we take these normal tissues, normal duct, and We deliberately stress them because we want to see how they respond to stress and whether we can drive them to become an early form of cancer. This is called DCIS, ductal carcinoma in situ. It is the most common early form of a non-malignant breast cancer. So we don't necessarily form DCIS, but we form stages leading to DCIS. So stress the normal tissue, how do normal tissues respond to that, and how can they recover? Yes, how can we reverse this? Now, one piece of data I'll tell you about is that my postdoc, Fernando Lopez-Diaz, discovered that a common protein that we all have that keeps our tissues healthy, called transforming growth factor beta, or TGF-beta, is a protein that normally helps our cells stay healthy. So when it recognizes damaged tissue, it helps the repair process. So that was known for a long time. It was also known that TGF-beta acts in very advanced cancers to enhance metastasis. But what Fernando found is that if he used TGF-beta on just slightly abnormal cells, or normal cells that had been damaged, or precancerous cells, it greatly improved their survival by impairing the damage response, and in so doing, greatly increased drug resistance of those cells. So that is shown here schematically. So now we treat the normal cells with damage or stress. In the presence of TGF-beta, we get greatly enhanced repair of the damaged tissue, or if the damage is too bad, TGF-beta will facilitate the death of normal tissue. If we treat slightly abnormal cells or precancers with TGF-beta, then we get impairment of our normal repair processes and enhanced survival now of these abnormal cells. It's really quite dramatic. TGF-beta greatly increases survival of both early cancers and metastatic cells. So the interesting thing is that TGF-beta itself has not changed, but the context of the cell in which it's operating in has changed. So we need to know what is it about the context that we might manipulate so that TGF-beta goes back to its protective role. Now, when we measure damage or stress, or if we measure any cancer cell, advanced cancer, early cancer, what are we looking for? Well, we're looking at not only what does the cell do? Does it die? Does it repair? Does it continue to live? We are looking at specific features of DNA, of chromosomes. Remember, your DNA is packaged into chromosomes, and proteins. And proteins, your DNA encodes proteins. So these are the major constituents of your cell that allows your cell to keep functioning. So we are looking at specific markers of DNA, chromosomes, and proteins. And just to tell you a little of our recent studies is that we are looking at changes in specific proteins upon stress that go back and impact chromosomal structure, which in turn alters our genes. And I'll just show you why that's important. Remember that our chromosomes, we have 46 chromosomes in a healthy human cell, and a hallmark of cancer is that these chromosomes become badly deranged. We suddenly have extra copies of some of our chromosomes or fewer copies of our chromosomes. We also have crossover events where one piece of chromosome joins another piece. So that's really bad. So your chromosomes are a mess in a cancer cell. So looking at a normal cell, remember that our chromosomes are normally highly organized into distinct territories. So a very tight control is maintained over the structure of your chromosomes. Each color is a different chromosome. You can think of this as La Jolla, Poway, Rancho Santa Fe, and El Cajon. So you maintain these nice tight territories by what are called molecular fences. You need some fences to keep the territories intact. And so your normal cell looks like this. You have lovely fences that keep one territory separate from another, and they have to be separate to maintain distinct functions. In a cancer cell, your fences basically collapse. And so we, instead of looking at the total collapse of all the fences, we are starting with early cancer cells and looking at a few fences that have collapsed. So how does fence collapse? How does loss of structural integrity begin? We are looking at this in early cancers, and we know a player, a protein that regulates fences. It's one of my favorite proteins. It's called CTCF. And we know that this becomes deranged upon stress and in cancer cells. And strikingly, we looked at normal breast cells, and we find CTCF, it's colored by green. There's lots of CTCF that completely fills the cell. It's keeping your chromosomes in nice territories. And then in abnormal breast cells, so not even cancer, but upon stress, prolonged stress, which creates a precancerous state, we see that CTCF is massively lost. And if I showed you a higher resolution picture, it would be redistributed in the nucleus. And so we think this is the beginning of the loss of the territories that leads to further instability of your chromosomes. And so our future directions are to investigate the events during cellular damage of normal cells that may lead to the generation and persistence of early cancers, determine how chromosome changes occur early in the genesis of precancers, particularly with CTCF and other proteins that hold our chromosomes together, and use the newly identified molecules as potential biomarkers and therapeutic targets to reverse early cancers and cancer progression. And I'd like to thank the members of my lab, Fernando Lopez-Diaz, Bettina Lehman, and David Henderson, who recently left to go back to Scotland. We collaborate heavily with the UCSD Moores Cancer Center and the National Disease Research Interchange to obtain our tissues. And our funding is largely from the National Cancer Institute and a program that actually puts physical scientists together with oncologists and molecular biologists. And I thank you for your attention.